fun.
Thank you. So, hi, my name is Paola Rubiano. I'm part of the group Predators and Toxic Prey, and I'm here this morning to talk about this particular insect called the milkweed bug, and how if he eats, therefore maybe he excrete. So I want you guys to meet the large milkweed bug. The name is Oncopaltus fasciatus, and it's been known as a research animal. Two different reasons. One of the reasons is that you can raise them and grow them in in several generations in lab conditions and study them through their own life history and different life stages, and allow them to understand different things of bugs. As example, you can understand their scent glands and how they accumulated here their pheromones and so on. Over here, you can also through their genome, you can understand how the evolution of the feeding ecology of bugs is been growing along the way. And on an interesting note, you can also use them as a model prey to understand the behavior of certain predators. But what I wanted to talk this morning is about one thing that I'm really interested in is that their connection through the diet. So these bugs are known to be host specific, means that they mostly eat certain plants of the family Asclepidae, especially genus Asclepias. And you can find them across North and Central America in different patches, both the bugs and the plants. They feed on the seeds, and the difference to other bugs that you can find crawling on the floor, they go up and up to the seed pods and show themselves in this colorful, bright way, and they feed on the seeds of this plant. And the reason that they have an interesting interaction is because of this substance that is going to be important for this talk, that is called cardenolites. So cardenolites, they can inhibit really essential enzymes to the functioning of the cells, meaning that they can cause real damage and real toxicity to a lot of animals that can feed on this compound. But because of this insect has really insensitive enzymes, they can eat it, and more than that, they can sequester. Sequester in this context means that they can eat it, accumulate it, don't die from it, and then in this particular case, secrete it to create an unpalatable defense when they've been eaten, when someone tried to eat them. This defense works in this way. As you can see them here in the nymph stages, they can have particular compartments to store a secretion, this liquid that is supposed to have cardenolites, but in the nymph stage, they don't have releasing points. The only openings are these dorsal areas that you can see them in the inside, that you can see filled with liquid. And they're supposed to have cardenolites around here, but there's not much information on the nymphs. The opposite happened in the adult stage, where it's been known that there's a space inside this area, the subticular compartments, where the defension, these liquid droplets rich in cardenolites, can be released through thin specialized areas. So when they are present, meaning they are being poked by a predator, these droplets are going to appear, and they're going to be really unpalatable and most likely toxic to whoever dares to poke them. But as I'm a chemist surrounded by biologists, my question is, what is the chemistry behind it? Which compounds can be released in this defensive secretion, actually, how they look, how they are made? Does the chemical profile of those secretion are going to vary according to the diet? And if do adults and nymph are going to show different profiles, because as we see, the droplets come from different areas. So with those questions in mind, I did something pretty straightforward in my lab. I raised this insect in sunflower seeds that, as you know, no toxicity there, and two species of the Asclepias. This one found in more tropical areas, this one more found in north and west, north and east parts of United States. But coming back to my title, if they're gonna secrete what they are eating, I need to understand their diet. So this is how a profile look. Basically, this picks tell me about the presence of a compound. And this peak contains way more information than this, contain the masses and the fragmentation pattern in a specific analysis tool. And through my analysis of that and isolation, meaning getting these pure compounds to understand it, I know that the main peaks are this type of cardinolites. The core is in these two is frugosite and gophrusite, which are really similar if you look at it, but the difference is that we have an hydroxyl here and we have an aldehyde here. And the two glucosidated version of these two molecules, meaning that they have an extra sugar, are gonna be the major two peaks. And the important part is that peaks that contain this aldehyde are actually not the major compounds. The major compounds is the one that have this hydroxyl. And when I tried to understand the other Asclepias, incarnata, I came across that it's a red trace. Incarnata is a more complex seed, it's a more complex food. They are eating not only the cardinolites that turns out to be actually the same composition, but they are eating way more things around here. So with that insight of what they are eating, it's time to see what they are secreting. So I pick some individuals, and through manual stress, I poke them and get these droplets out of them. And in an interesting finding, turns out that most of that secretion is quite oily. It's made of unsaturated triacylglycerol. They basically cover themselves in oil, up to 20%, 24% in the case of adults, and 50% in the case of nymphs. Which sounds fun, but for me, I had to get rid of it because it interferes with all my analysis. So I had to do some different paths to remove it and get it more clear to do a more profile definition. So here I will look at the defensive exudates of the nymphs in these three seeds: sunflower, Asclepias incarnata, and Asclepias curasabica. What I can find is that they managed to get these two molecules out of the sunflower seed for the defense. I'm still working on what these molecules mean. The only thing that I can say is that I did not find an amount of cardinolites in the nymphs of incarnata that it could be detected by the equipment. There's just not much presence there. But in the case of the nymphs raised on Asclepias curasabica, I did find cardinolites, and more interestingly, I found that the big peak is frugosite, not the glucosidated version that it was quite available in their diet, meaning that they were quite smart and took the sugar out of it to their own source of energy and enrich the compound that they're gonna use for defend themselves. And in this particular peak, I call it a modified frugosite because although it has the same patterns and a lot of information that is inside it differs the mass meaning that they took this molecule and they added something extra, maybe to able to transport it more effectively or something like that. But what I can say is that so far, this is my question: what are those molecules that they got it from the sunflower seeds? What does this mean? That if the goal is eat and secrete, the winner was curasabica because allowed them to get a good diet, to get them frugosite that they want to defend themselves, and they also get creative. And incarnata, they didn't manage to sequester anything, at least in not an amount that is significant. And when I did the same for the adults, I also find these molecules here that they got from the sunflower seeds. We had a clear part that there's no cardinolites because no one has died from eating sunflower seeds. And this, interestingly, in the case of adults in incarnata, they did manage finally to get enough cardinolites to be detected and present. But again, curasabica, by having a more easy available cardinolites for them to obtain, it has a more clear presence here. And we see again frugosite and gophrusite being the major compounds, so meaning that they are really keen on getting the glucose out of these compounds. And as I mentioned before, the relationship on size between frugosite and gophrusite is different from what they eat it, meaning that there seems to be keen on enriched and accumulated gophrosite over frugosite. And my theory is that gophrosite, by having an aldehyde had a reputation of being nasty and reactive. So if they want to defend themselves, or if they have really sensitive transport enzymes or something, they want to target a predator to say, hey, don't eat me, they are accumulating aldehydes. So again, we see that I'm still wondering what those molecules are. The winner is going to be, again, Curasavica. They get in a lot of sugar, and they seem to get a lot of gophrosite, that they have this particular attraction. And at least this time we realized that adults being at least more adapted to accumulated in a better way cardenolites, they managed to get enough cardenolites for me to detect it. So they actually getting something out of a complicated diet. Now, what I've been working is trying to do a preliminary statistical analysis where I can try to understand how these defensive exudates correlate with each other, how they're different, how they are similar. And so far, what I can understand is that the defensive exudate of Anoncopeltus raison curasavica differentiate from all these other variation of life stages and diets from the high amount of the cardinalize that is there. And when you try to cluster all these defenses together, you see a clear definition that adults differ highly from NIMS defensive exudates. But what it strikes me as surprising is that you would expect that Asclepias race bugs share more characteristics, but turns out that because of the lack of cardinolites present in the defensive exudate, like the really low amount present in the defensive exudate of Oncompeltus raison incarnata, makes it more similar to sunflower seed defensive exudate, almost a non-toxic bug, a non-toxic diet. That so it's more prevalent, the fact that they don't have enough cardinolites and they don't cluster with a diet that is actually essentially the same genus, which is slightly different. But to understand this beyond the chemical point of view, my colleague is trying to see, okay, but what does this mean? And what this mean is how a predator can learn and react if it's encountered a bug that it was in the patch full of incarnata versus a bug that it was in a patch that it was full of curasavica. And if those bugs intrinsically are being more or less defended, meaning that they are gonna avoid the getting eaten, or the predator is gonna see that it has a nasty bug in the mouth and throw it away, or how is the learning process for a predator? So these are part of also of our group, our nice mantids. So with that, I wanna say thank you very much. I wanna thank you, my supervisor, Hannah Rule and Kristen Patz, and my friends and colleagues from the Predator and Toxic Prey. And I am PRS Masplank, and so much, thank you so much to the greenhouse team to grow up so many Asclepias plants for my research. So, danke, thank you, and gracias.